This lecture is going to continue off of our first Chapter 4 lecture on cells and organelles. This one will focus mostly on the distinct organelles and the basic characteristics of each. The first one up is the plasma membrane. So here showing the outside of our eukaryotic animal cell and the membrane that's surrounding it. Remember, this is a phospholipid bilayer with the hydrophobic inside and hydrophilic heads on either end. So if we pretend this is the inside of the cell down here, there's a hydrophilic head interacting with the cytosol, and then this is the outside of the cell here, and again a hydrophilic surface interacting with the aqueous environment that the cell is in. There are other things besides lipids in your plasma membrane. One might be proteins. So here we have a protein. It's got a bunch of alpha helices in the middle. These alpha helices are hydrophobic, so they don't mind being in here in the middle with these lipids, the hydrophobic tails. The ends, however, are hydrophilic, so they don't mind, again, being on that end. And that's what helps this protein stay localized here in the membrane, or those two characteristics. There may also be other things on the outside of these proteins, such as carbohydrates, and those are usually involved in some sort of signaling. Next up is our nucleus. So remember, this is the site where all of the DNA is. Here we've got this dark spot in the middle, the nucleolus. This is where ribosomes and ribosomal RNAs are made. This is the outside of the nucleus, as seen with the electron microscope. You can see all these little indentations, kind of looks like a golf ball. This nucleus is covered with pores, so there are openings in it where things can get into and out of it, and we need that so proteins that are involved in gene expression and replication can actually get into that nucleus, and that mRNAs that are made can actually get out so they can be translated in the cytosol. Those are really tightly regulated. They're like these little cage-like protein complexes that allow things in and out. Now up here, I'm showing you guys a picture from a journal article that was published. This is a nucleus where they have tagged each one of the chromosomes, you can see them numbered over here, with a separate fluorescent tag so that they can tell where those chromosomes are at. This is really amazing because this nucleus is very highly organized. You hardly see at all any areas, if you look over here, where these things overlap. So there's a very specific area where, say right here, this is chromosome 9. This is where all of chromosome 9 is and where it stays. Now remember those X-like formations that chromosomes make? Those are only seen during mitosis or meiosis. So that's only seen when your cells are replicating. So most of the time, your chromosomes are all spread out like this, like a big network, so that they can actually be used for transcription. So this is very cool that they're tightly organized, and that would be really important when the cell goes to replicate that DNA, that all of that chromosome 9 is all right there and it's not tangled up over here with this other chromosome. Next up, mitochondria. This is where all the energy is going to be made in your cells. So remember aerobic respiration, where you have glucose, a sugar, oxygen, and enzymes, and you're going to make carbon dioxide, water, and ATP with that. We'll get into that more in the next unit of how exactly that happens, but for now just remember that this is the overall process and the reactants and the products that you get from that. And this involves the oxidation of sugar and other molecules. Ideally, your mitochondria like to use sugar. When sugar is not available, it will use proteins and fats for this process. Another aspect of the mitochondria is the mitochondrial DNA. There's a specific piece of DNA only found in the mitochondria that codes for enzymes that are needed in the mitochondria to do this process. Not all of those are coded in that, so a lot of them are actually in the nucleus as well, but some of them are on that mitochondrial DNA. And remember that all of your mitochondria only come from your mom, so the mitochondria in the egg cell are what get passed on to the offspring. Remember, your mitochondria have a membrane around the outside. There are actually two of them. There's one that goes all the way around the outside, and then one around the outside, but it also has these invaginations in the middle called cristae. So it has these pieces that go throughout the middle and increase the surface area. So remember, we just talked about surface area and how that's important. So by increasing the surface area in the middle, we can increase the number of enzyme complexes. So your electron transport chain, for example, is found localized in this membrane right in here. So we can increase the number of those that we have on those membranes to make more and more energy. The number and the location of your mitochondria are going to be different depending on what tissue you're in. So something that needs a lot of energy, such as your muscles or your heart or your brain, are going to have more mitochondria than something that doesn't. And the location of those are going to be varied. So we'll look at some pictures later on that show that your mitochondria in your muscle cells is localized so it can feed those muscle cells appropriately. Here's a YouTube link, so I suggest you go and take a look at that, particularly at video 1. Your book and a lot of literature shows mitochondria as just these kind of oval pieces in your cell that are actually very dynamic. So they make these huge networks of very, very long mitochondria, and they will divide and attach to each other off and on constantly. And people that generally exercise quite a bit tend to have more fused mitochondria. So they will have longer mitochondria, and those are generally thought to be more efficient and work a little bit better than mitochondria that are just in this kind of formation. So sedentary people generally have mitochondria that are small and divided more like you would see in this cell. So it's something just to keep in mind. Chloroplasts, only found in plant cells, and these are going to do the opposite of the mitochondria. So they're going to take solar energy, so light from the sun, and carbon dioxide, and they're actually going to make sugar and oxygen, whereas your mitochondria are going to take sugar and oxygen and break it down into carbon dioxide. Chloroplasts are part of a big group of organelles called plastids. Plastids generally contain pigments or food. So in this case, they're going to be making nutrients, but they also contain pigments, which are the green pigments that are what make give plants their green color. They've got two membranes around the outside, just like mitochondria do, and they've also got an inner membrane system with these stacked discs. So each one of these little discs is called a thylakoid. If you stack a bunch of them together, they're going to call it a granum. And these are where all the light reactions for photosynthesis are going to take place. There's a theory called the endosymbiont theory of how we got these two groups of prokaryotic and eukaryotic cells. And the general idea is that we had some sort of cell that was similar to a eukaryotic cell but didn't have mitochondria yet, and a bacterium got absorbed into the cell and they begin living symbiotically together, and those then became mitochondria. So several things support this theory. One, that mitochondria have their own DNA. Two, that they have their own energy production. This process actually still happens. So there are protozoans that have been incorporated into other protozoans and now live symbiotically in this kind of manner. And it's thought that mitochondria and chloroplast actually did this at two separate times. So this made, our animal cells, and then incorporating the chloroplast then led to all the plant cells. The fifth organelle actually contains several organelles within this. So we're going to call the overall thing the endomembrane system. And then the first part of that is the endoplasmic reticulum. So endoplasmic just meaning in the cytoplasm, and reticulum meaning network. So it's this network of membranes that we find in the cytoplasm. So they have tubular membranes and then what we kind of call flattened stacks. So it's kind of this like kind of pancake looking thing. And the outer membrane is actually continuous, so you can see it connects here with the nuclear envelope. So things can get passed back and forth, proteins can get passed between these two. There are two components, the rough endoplasmic reticulum, and it's called rough because it has all these ribosomes, all these spots, see over here, on the outside. And those make proteins that are intramembrane proteins, so all the ones that would be within the membrane like we saw in that plasma membrane slide, and also proteins that are secreted. And we make them here so then they can be right away incorporated into a membrane. We don't have to go through a process of trying to put them in there once they get to the membrane. And here's electron micrograph. You can see all those little spots are all the ribosomes. The other component is the smooth endoplasmic reticulum, and its job is to make lipids and steroids. It also is involved in detoxification of drugs. So here you can see the smooth. It doesn't have the ribosomes on it like the rough does. The next component of the endomembrane system is the Golgi apparatus. So again, we have stacks of flattened vesicles, so again, this pancake-like thing. Now these aren't all connected, so they're stacks on top of stacks on top of stacks. And these pieces that break off of it can actually go either direction, so they can actually come into the cell or they can go out of the cell. It's involved in processing and packaging secretory proteins, so proteins that are secreted. There are other modifications that have to be made, so sugars that have to be added onto the outside of them and that sort of thing, and that's what the Golgi apparatus is going to do. It's going to process those, put those sugars on, and then send them wherever they need to go. That might be to another organelle like the mitochondria, or it might be completely outside of the cell to send to another cell. And here you can see that process. You can see those proteins coming from the endoplasmic reticulum, merging with the Golgi apparatus. Now those sugars can be added. They're going to travel through several layers of the Golgi apparatus while they're being processed and come out the other side, and then they can be secreted outside the cell or to another organelle. And likewise, a vesicle can come from outside the cell and go the other direction, and we call that retrograde signaling. So it's going backwards and sending some sort of signaling or some sort of proteins into the cell. Our last component of the endomembrane system is a lysosome. These are round organelles, and they don't really have much distinctive features on the inside here, so you don't really see anything distinctive, just a round organelle. These are used to store enzymes that digest other molecules. These can be waste type molecules, or it can be molecules that weren't made properly. So like a protein that was truncated and not finished, it would be sent here and then broken down into the amino acids, and those amino acids can be sent back out to be used in other things. Now, why would we need an organelle to actually store those digestive enzymes? Well, the reason would be if you had those floating out in the rest of your cell, those enzymes don't know which parts that they're supposed to digest and which parts they aren't, so they would just be tearing everything down. So we contain it here in an organelle, and then it only digests the things that come inside of it, the things that are supposed to be digested. There's even a protective coating around the inside of the lysosome so that those enzymes don't digest the membrane of it as well. Another function of the lysosome is autophagy. This is the recycling system of the cell. So like I mentioned, anything that wasn't made properly can be sent here and broken down, and they can be recycled and reused in another way. All right, our next organelle, we're out of the endomembrane system now. Our next organelle is a peroxisome. So again, these are small and round organelles, and they don't have much structure. So very similar kind of overall look to a lysosome. There are several different functions of these. One is generating and degrading hydrogen peroxide. So hydrogen peroxide can be produced as a byproduct of respiration, and it's not good to have a lot of this hanging around your cells. It's going to start to damage different molecules. So there's an enzyme called catalase. It's in these peroxisomes. It's going to degrade that hydrogen peroxide. Now, some hydrogen peroxide is good. It can be used for signaling. So you don't want to get rid of all of it, but you want to get rid of most of it. It can also degrade things like methanol, ethanol, so any alcohols, formate, formaldehyde. All these things, again, byproducts of processes that go on in your cell and waste products that your cell needs to get rid of. Your peroxisome is going to be involved in doing that. It can also break down large fatty acids. This is before the mitochondria can do it. So the really big ones your mitochondria can't break down. It'll break them down, and the peroxisome will break them down into smaller pieces, and then your mitochondria can handle the rest. There's a very high concentration of peroxisomes in your liver and kidney. And if you know anything about the functions of those organs, that's your detox system. Those are the things that are going to clean the waste products out of your blood and out of the rest of your body. So they've got a very high concentration of peroxisomes. But peroxisomes are in every type of cell that's in your body. Obviously, if you drink alcohol, so ethanol is going to be in there, and your liver and kidney are what's going to break that down. If you've ever heard about juice cleanses and that sort of thing, I will tell you those are complete crap. You do not need a juice cleanse to get rid of things to detoxify your body. Your peroxisomes and your liver and kidney are what's going to do that. Juice is not going to do a thing to detoxify you. So if you want a cleanse or detoxification, what you need to do is drink a lot of water, eat your fruits and veggies, and your peroxisomes and your liver and kidney will take care of the rest for you. Number 7, vacuoles. So again, we're back to our plants. These are not found in animal cells, at least not the big ones. We do have small ones in animal cells, and those are mostly temporary storage and transport. They're not playing a huge role in our animal cells like they are in our plant cells. In protozoans, so protists, they feed by endocytosis, so by taking in vesicles, just like we talked about with our endomembrane system, taking in vesicles, they're going to feed that way. So they're going to engulf a vesicle or a vacuole and then merge those vacuoles with a lysosome that then is going to break down those components that are inside. And then our plants. The vacuoles are supplying turgor pressure to our plants. So again, if you have a wilted plant, it's because there's not enough water in this vacuole, and then it's making this cell kind of collapse in on itself. So it's maintaining the structure of the plants. The cytoskeleton is a structural component of your cells. It's made up of highly structured proteins. So in this picture, all of the green that you see are cytoskeleton proteins. They're long filaments that give the cell its 3D shape. They're very dynamic in that they aren't just sitting here as they are. From one end to the other, they grow on one end and they degrade on the other. So in videos, you will see it looks like they move, but they don't actually move. It's just that one end is growing and one end is degrading. So they're dynamic in positioning themselves to where they need to be and where they need to go. Like I mentioned, they help with cell shape. They also help cells move. So at the end of this lecture, there is a video you can watch, and it shows a white blood cell that has very distinct movement. So it's the cytoskeleton that's helping it do that. They are active in cell division. So they're the pieces that attach to the different chromosomes and actually pull them apart during cell division. And they can actually move organelles. So there are motor proteins that can attach to the cytoskeleton filaments, and they kind of walk along the filament, and they can drag an organelle with them if those organelles need to be moved to another part of the cell. There are 3 types of cytoskeletal filaments. One is microtubules. Those are the biggest. Obviously, these intermediate filaments are going to be in between, not the biggest, not the smallest. And microfilaments are the smallest. You might have heard of actin. It's probably the most common one. and those are the smallest ones. They each have different functions within the cytoskeleton. The extracellular matrix is the last thing we're going to talk about. It provides support around cells, around the outside of cells. So in plants, this is the cell wall. That's made primarily of cellulose, and that's going to provide this support around the outside of the plant cells. In animals, it's collagen, which is part of your connective tissue, and it's very elastic, very flexible, so it's going to give your organs and your tissues that flexibility that they need to be able to move. Also, within the extracellular matrix, you find junctions between cells. So there are ways that cells can connect so they don't have to use endocytosis or exocytosis to transport things. They can use these junctions in between cells, and we'll talk about those more a little later on in our class. Now, this last slide has a link to a video. I suggest you go and watch that video. It's several years old now, but it's still very beautiful. So it's not high def, so forgive the standard definition, but it was made by a group at Harvard. It's a really gorgeous animation just showing a lot of dynamics and what's happening in the cell. And this is the narrated version, so they're going to tell you about some things that are a little more in depth than what we're going to talk about, but it'll be interesting for you, I think, to see these things kind of play out.